This is a lecture on Introduction to Biostatistics by Department of Community Medicine, International Medical University. Let's start with the meaning of biostatistics. By definition, it is defined as the application of statistical principles in medicine, public health, or biology. These statistical principles are based in applied mathematics, which help us in developing tools and techniques for data collection and summarizing. Not only that, statistical principles are used for analyzing and interpreting the results from which we can make inferences and draw conclusions. Here is the list of areas where you can apply statistical principles. 1. Collection of data. Let's say we want to find out the vital statistics to inform and monitor the health status of a population. An example is mortality rate, where we want to find out how many people die from a particular disease. In such case, we need to start with collection of information or data. 2. Analysis of data. Example, we want to find out what time of the year that road traffic accidents occurs the most. We will need to look into the accidents records and analyze the data to identify when these accidents happen the most to implement safety measures at a particular time. 3. Clinical trials. Example, we want to find out if a new antihypertensive drug performs better than the standard treatment. We can perform a clinical trials and analyze the data obtained from these trials, interpret them to make conclusions to say if the new drug is better or not. 4. Surveys. Example, to estimate the proportion of low-income women who have given birth. 5. Studies. This will help us understanding the causal factor or factor associated with a health outcome. Example, investigating whether or not an exposure to electromagnetic field is a risk factor for leukemia. This is the picture showing the data view of the data collected for a study on SPSS statistical analysis software. It will be very difficult for us to handle this if we don't know the underlying statistical concepts. The cells in each row of the data sheet represents the different characteristics of a participant in your study. And the participant in your study can be as many as thousands. For us to be able to handle this, we need to know what are the tools and techniques involved in data collection. What are the systematic ways to represent the data we obtain? What are the concepts we need to know to analyze this data? And eventually, how are we going to interpret the results of the statistical analysis? That is why, in a typical statistical process, the following 4 stages are needed to go through. 1. Collection of data. 2. Presentation of data. 3. Data analysis. And 4. Interpretation of the results. The next step we need to know is the fundamental statistical methods. There are 2 statistical methods, which are descriptive statistical method and inferential method. So let's move on to see what they mean. Descriptive statistic. It is a set of observations that describe the characteristics of a sample in our study. However, it does not make any conclusions from the statistical tests we made. It is used for informational purpose only and not to make conclusion on whether an exposure is a risk factor of a health outcome. We can summarize and display the data numerically or graphically. For example, we can use mean, median, mode, standard deviation, etc., or even using diagrams such as bar charts, histograms, box plots, stems and leaves to visualize the distribution of data, identifying the patterns which can eventually lead us to hypothesis generating. This is the example how we can visually display the data to describe how the findings looks like. Diagram A tells us the proportion of people in each level of obesity and age group who have diabetes. Diagram B tells us the frequency of age group for 40 patients in an intensive care unit at a hospital. Here are more examples to graphically describe your data. Figure C shows the distribution of diastolic blood pressure in a population we study. It divides the data into quartiles. Figure D is an example showing the health expenditure as percent of GDP for countries from 1960 to year 2000 in a line graph. The next type of statistical method is inferential statistic. It uses data from a sample to make comparisons and draw conclusions about a larger group that the sample represents. In another term, it is a procedure for making inferences or generalization about a larger population from a sample we draw from that population. The reason is that it's not possible for us to study each and every one of an entire population. It helps us in estimating population values from sample values. We use inferential statistics to distinguish whether the differences we found in a study is a true difference or it is a variation that occurs by chance. It also allows us to test the hypothesis we develop for our studies. This is a diagram showing how samples are selected from a larger population to study and to draw a conclusion about the same populations the samples are drawn from. Regardless of whether it is finite population, where we can count, or infinite population, where we cannot count, it's not possible to obtain the data from all the members of the population. It is good to pick up the random samples from the populations to calculate the sample statistic in order to obtain the population parameters. Take note of the term sample statistic and population parameters, which will be explained in the next two slides. In this diagram, you will also notice that statistics that are calculated from the samples are called sample proportions or sample mean. In order to obtain the true proportion and true mean of the population, it is required to make estimation for population parameters to describe or generalize your results into population. So, what are the ways to generalize from samples to populations? Commonly used statistical methods are using confidence intervals to make estimation about the population or using test of significance to test the hypothesis we develop for our study. These methods will help us in making decisions for the population characteristics. The information we need to learn from this slide is the terms that are used for population and sample. For example, symbols for mean and standard deviation for population and sample are different even though they have the exact meaning. Mu and delta are used to represent population mean and population standard deviation respectively, and x bar and small s for sample. Please take note that we use the word parameter for numerical values that summarize all the data of population, and we use the word statistic for the sample data. These are the terms and symbols used for population parameters and sample statistic. Example, mean for population parameter is mu, and that for sample statistic is x bar. Standard deviation for population is delta, and that for sample is capital S, and so on and so forth. This table summarizes the basic statistical terms for population, sample, parameters, and statistics, the meanings of which are explained in the previous slides. The two new terms we will have to learn here are variable and data. Variable is the characteristics measured on individuals drawn from a population under study. It can be their age, their gender, height, weight, etc. On the other hand, data is the measurements of one or more variables made on a collection of individuals. Example, their age values, height values, or weight values. So let's go more into details about data and variables. Data is regarded as a key component of a study. It is important as different statistical methods are used based on type of data. Obtaining a good data set is crucial as we cannot have a good study without good data. There are two types of data or scales of measurement. One, quantitative or numerical data to qualitative or categorical data. Therefore, the next step is to find out what are quantitative and qualitative data. Quantitative data are numerical data, which can be subdivided into continuous and discrete. Continuous data are values on metric scale. Basically, they can be measured. For example, age in years, height in meter, blood pressure in millimeter mercury, hours, degrees Celsius for body temperature. Discrete data, on the other hand, are integer values which are counted units. For example, number of siblings, number of children, number of times visited to a clinic, number of cigarettes one smokes per day. Qualitative data are called categorical data in another term. There are three types of categorical data: binary, categorical, and ordinal. Binary data are used to classify participants as possessing or not possessing a particular characteristics or attribute. For example, disease presence or absent, male or female, treatment or no treatment. Nominal are values in arbitrary categories and they have no unit. Example, race, blood groups, types, educational level. Ordinal are values in order category. It has intrinsic value, but they have no units. For example, BMI, score, grades, and age group. This flow diagram summarizes the type of data that are needed to know. One thing important to note here is that same property can be measured in different scale. For example, age in year is the continuous numerical data, but if you group all the age into young age group, middle age group, and elderly group, it becomes the nominal data. This flow diagram helps us in deciding the type of variables we are dealing with. First is to see if the variables have units. If not, we can put them in qualitative type of data. Next step is to see if the data can be put in meaningful order. If yes, they are considered ordinal data, and if not, they are considered nominal. So let's go back to the top of the flowchart. If the variable has a unit, it is put in quantitative type of data. Next step is to find out if the variable can be measured or counted. If it is measurable, it is considered as continuous metric data, and if it can be counted, considered as discrete metric data. Variables. Variables are the characteristics of the units measured or observed. Next is the variables. Variables are the characteristics of the units measured or observed. There are two types of variables. One, dependent variable, which is also known as outcome variable, and two, independent variable. Example, in a study of hypertension among 500 school children, investigator measured systolic blood pressure and diastolic blood pressure for each pupil. Pupils' age, height, and weight are recorded in addition to systolic and diastolic readings. So here are the questions. How many variables are there? We will find 5 variables. And what are the dependent variables? Systolic blood pressure and diastolic blood pressure, as these are the outcomes the investigator wishes to measure. What are the independent variables? Age, height, weight, as these are the variables the investigator wishes to find whether they are associated with low or high or normal blood pressure in 500 school children. This table describes the types of variables collected in a case control study. This study collected data on age, ethnicity, social class, number of children, menopausal state, and body mass index. From here, we can see that Age is the continuous data. Ethnicity is the nominal data. Social class is the ordinal data. Number of children is the discrete data. Menopausal state is the nominal data. And BMI is the continuous data. This is the end of the lecture on Introduction to Biostatistics. Hope you have proper understanding on the statistical methods, differences between population and sample, and type of data and variables. Thank you.